The universe contains billions of habitable environments and has existed long enough for civilizations to spread across galaxies. Yet we observe absolutely nothing. No signals, no megastructures, no evidence of life. Economist Robin Hanson modeled this silence as a mathematical necessity. If the universe can generate life, but none is observed, an evolutionary bottleneck must exist, a statistical barrier that stops dead matter from becoming an interstellar civilization. He called this the Great Filter. Hansen mapped this trajectory across 9 stages. It starts with a star system with organic molecules, moves through single-celled organisms, sexual reproduction, and climbs towards space colonization. Steps 2 through 6 represent the biological hurdles, the transition from raw chemistry to complex multicellular biology. Our survival as a species depends on locating exactly where this barrier sits. If the hardest evolutionary hurdle is in our past, we have already survived it. If it lies in our technological future, our extinction is a statistical probability. We no longer have to rely solely on theoretical models. Observatories like the James Webb Space Telescope allow us to physically test the filter hypothesis. When an exoplanet passes in front of its host star, a fraction of that starlight filters through the planet's atmosphere. Specific molecules absorb specific wavelengths of light, leaving dark gaps in the spectrum that reaches our detectors. By capturing this transmission spectrum, we are directly checking the universe for Hansen's biological steps. We are looking for the atmospheric exhaust of alien biology. Detecting these biosignatures provides the data required to place hard observational constraints on exactly where the Great Filter resides. Before we scan a spectrum for biology, we must first locate environments physically capable of sustaining it. Astrobiologists define the habitable zone as the precise orbital annulus around a star receiving the correct stellar flux to maintain surface liquid water. This model requires a rocky composition with a carbonate silicate thermostat and an atmosphere dominated by nitrogen, carbon dioxide, and water vapor. This chart displays baseline atmospheric habitability markers. To prove a planet has the right surface pressure and temperature, we look for spectral absorption lines for water and carbon dioxide. On the right, collision-induced absorption reveals how nitrogen molecules colliding under high pressure create temperature-dependent absorption features. Confirming water, carbon dioxide, and atmospheric density verifies the planet's structural capacity for life. Only then do we analyze the spectrum for biological anomalies. To predict what living planets will look like, researchers combine 2 computational tools. Photochemical models calculate how stellar ultraviolet radiation drives chemical reactions in an atmosphere. Those results feed into radiative transfer models, which calculate exactly how that gas mixture will absorb and scatter light. This modeling reveals gaseous biosignatures. On Earth, oxygenic photosynthesis harnesses water and carbon dioxide, continuously outputting molecular oxygen as a biological waste product. Stellar ultraviolet light breaks apart this oxygen in the stratosphere, reforming it into ozone, which leaves a much stronger, more easily detectable signal. This synthetic radiant spectrum of modern Earth maps wavelengths absorbed by our biosphere. We identify the massive ozone absorption trough near 9.6 micrometers and the sharp oxygen A band at 0.76 micrometers. The most revealing feature emerges when identifying methane alongside these oxygen markers. Methane and oxygen are highly reactive. In a static environment, they rapidly oxidize and annihilate each other. Finding them coexisting indicates extreme chemical disequilibrium. A biological engine must actively and continuously pump them into the air, or the methane would disappear entirely. Atmospheric gas is the most accessible metric, but biological structures and global cycles also leave distinct fingerprints. We classify these as surface biosignatures. The primary example is the vegetation red edge. Photosynthetic structures face a physical constraint. They must absorb visible light to generate energy, but must reflect near-infrared radiation to avoid catastrophic overheating. This dual requirement creates a steep step function in the reflectance spectrum, an optical signature that raw geology does not produce. We can also observe how a biosphere operates over time by measuring temporal biosignatures. As a planet rotates through its seasons, plant matter blooms and decays. This causes massive cyclic shifts in atmospheric carbon dioxide concentrations and planetary albedo. The entire atmosphere oscillates in response to the orbital mechanics. The vegetation red edge, seasonal chemical oscillations, and extreme atmospheric disequilibrium, finding these simultaneously drops the probability of a non-biological false positive to near zero. We have found a living world. This astrobiological framework maps directly back to the Great Filter. Researchers Jacob Hack-Misra and Edward Schweiderman proposed using these exact parameters to calculate our existential risk. Consider the first potential outcome from our telescopes. We scan the galaxy and find widespread evidence of chemical disequilibrium and vegetation red edges. This proves the transition from dead matter to complex biology is common. Now, pair that with our current radio telescope data. We observe 0 technosignatures, no atmospheric industrial pollutants, no radio traffic, and no Dyson spheres. If life easily evolves to a complex state but never colonizes the stars, the mathematics point to a single conclusion. The evolutionary bottleneck sits right in front of us. If we find a universe packed with biology but entirely devoid of engineers, the act of developing advanced technology carries an insurmountable risk. Civilizations routinely emerge, reach our level of industrial capacity, and reliably destroy themselves before they can spread. But there is a second outcome. We scan thousands of exoplanets in the habitable zone, and the data returns completely flat. Every atmosphere we sample rests in perfect, dead chemical equilibrium. If biology is statistically absent from the universe, the barrier sits firmly in our past. It means the transition from sterile chemistry to self-replicating cells, abiogenesis itself, is a supreme statistical anomaly that almost never occurs. Counterintuitively, a sterile, empty sky is the most optimistic result we could ask for. It means Earth has already cleared the hardest mathematical hurdle the universe provides. We are the rare exception. This rigorous cataloging of atmospheric chemistry serves as a diagnostic map for our own species. It provides the only mathematical tool we possess for predicting whether human civilization is destined to persist or disappear.